Let's take a moment to introduce glands. Glands can be unicellular, multicellular, or even organs. There are two main categories for glands, exocrine or endocrine. I will briefly introduce endocrine glands here. Endocrine glands are ductless glands that release their secretions into the surrounding extracellular fluid. That is, endocrine glands release their secretions into the tissue fluid that surrounds them rather onto an epithelial surface. More specifically, endocrine glands produce hormones that are chemical signals. These chemical signals signal distant target cells or organs. A target cell or organ has a receptor that can bind to the hormone. Once bound, the hormone causes the target cell or organ to change its activity or physiology. The endocrine system will be discussed in more detail at a later date. For today, let's focus on epithelial glands. Many epithelia contain gland cells that produce secretions. Exocrine glands discharge their secretions onto an epithelial surface using ducts. This surface can either be the surface of the skin or into the cavity of the mouth or along the digestive tract or even onto the surface of the lung. All of these locales have an epithelial surface. Exocrine glands are classified by the type of secretions released, the structure of the gland, and the mode of secretion. I will not discuss the structures of the glands, for example, if they are tubular or alveolar, but I do want you to know that exocrine glands can be either multicellular or unicellular, secrete mucins, enzymes, water, and waste products. Most importantly, these secretions are released at the apical surface of the individual gland cells. Epithelial gland secretion is the process whereby gland cells obtain needed substances from the blood and transform them chemically into a product that is then discharged to the exterior of the cell. More specifically, the protein product is made in the rough ER, then packaged into secretory granules by the Golgi apparatus, and ultimately released from the cells. At the top of this slide, I show you an example of a unicellular exocrine gland. It is the only important example of a one-celled exocrine gland. It is a goblet cell. A goblet cell is shaped like a goblet, a drinking glass, with a stem. Goblet cells are numerous in the epithelial lining of the intestines, and I show you them on the photomicrograph next to the cartoon drawing of a goblet cell. First, the arrow points to what looks like a purple outlined jelly bean. These are really called intestinal crypts, and you will become more familiar with these crypts when we learn about the digestive system. Now look more closely at this crypt and find what looks like white bubbles. What you are really seeing is the clear mucin within the superior portion of the goblet cell. It is colored orange in the goblet drawing. Upon even closer scrutiny, you can see that below the white bubble, you see a dark staining nucleus. In fact, the nucleus of the goblet cell is found in the stem portion of the cell. An example of a multicellular gland is shown at the bottom of the slide. Each multicellular exocrine gland has two basic parts, an epithelium wall duct and a secretory unit consisting of the secretory epithelium. The photomicrograph at the bottom of this slide is a typical sweat gland. Exocrine cells may be categorized according to the nature of the secretion produced. Serous glands secrete a watery solution that usually contains enzymes, such as the salivary amylase in saliva. Mucus glands secrete glycoproteins called mucins that absorb water to form a slippery mucus, such as the mucus in saliva. Mixed exocrine glands contain more than one type of gland cell and may produce two different exocrine secretions, one serous and the other mucus. The submandibular gland, one of the salivary glands, is an example of a mixed exocrine gland. A glandular epithelial cell may use one of three methods to release its secretions. Merocrine, apocrine, or holocrine. In merocrine secretion, the secretory product is released through exocytosis. Review your notes from Chapter 2 on what exocytosis means. This is the most common mode of secretion. Goblet cells are a perfect example for merocrine secretion. Apocrine secretion involves the loss of cytoplasm as well as the secretory product. The apical portion of the cytoplasm becomes packed with secretory vesicles before it is shed. So it is a lot like exocytosis, but some of the cytoplasm is lost. Lactiferous, or mammary glands, are an example of this type of secretion. With glands that use either merocrine or apocrine modes of secretion, the process leaves the nucleus and Golgi apparatus of the cell intact, so the cell continues to live and secrete. Holocrine secretion destroys the entire gland cell. During holocrine secretion, the entire cell becomes packed with secretory products and then bursts apart. The secretion is released and the cell dies. Further secretion depends on gland cells being replaced by the division of the stem cells. Sebaceous glands associated with hair follicles produce a waxy hair coating by holocrine fashion. Connective tissues are found throughout the body but are never exposed to the environment outside the body. All connective tissues have three basic components. 1. Specialized cells. 2. Extracellular fibers. 3. A fluid known as the ground substance. The extracellular fluid and fibers create the matrix that surrounds the cells. Connective tissues have many different functions, which include establishing the structural framework for the body, transporting fluids and dissolved molecules from one region of the body to another, protection, interconnecting other tissue types, storing energy, particularly in the form of lipids, and defending the body from infection. It is important to know that no single connective tissue type performs all of these functions. Rather, there are many different connective tissues, each with its own unique characteristic and function. All connective tissues arise from the middle layer of a developing embryo called the mesoderm. Mesenchyme is a word that describes stem cells that respond to local injury or infection by dividing into daughter cells that differentiate into fibroblasts, macrophages, or other connective tissue cells. Basically, think of mesenchyme as mesodermal cells that roam the body and can use gene expression to direct what type of connective tissue they will become. There are three main categories of connective tissue, and these are listed at the top of the slide. Connective tissue proper refers to connective tissue with many types of cells and extracellular fibers suspended in ground substance. These connective tissues may differ in terms of the number of cell types and ground substance, and I will discuss the different types that fall into this category shortly. The second type of connective tissue is considered to be supportive. This type of connective tissue has a less diverse cell population than connective tissue proper, and a matrix that contains closely packed fibers. There are two types of supporting connective tissues, cartilage and bone. Fluid connective tissues have a distinct population of cells suspended in a watery matrix that contains dissolved proteins. There are two types of fluid connective tissues, blood and lymph. This is a handy little cheat sheet that shows you the three main categories of connective tissue and some of their characteristics. Connective tissue proper contains extracellular fibers, a viscous or thick ground substance, and two classes of cells. Fixed cells are stationary and are involved primarily with local maintenance, repair, and energy storage. Wandering cells are concerned primarily with the defense and repair of damaged tissues. The number of cells at any given moment varies depending on local conditions. Fixed cells include fibroblasts, fixed or tissue macrophages, adipocytes, mesenchymal cells, and, in a few locations, melanocytes. I will not discuss melanocytes here. Rather, I wait for the integument chapter discussion. Fibroblasts are the most abundant fixed cells in connective tissue proper and are the only cells always present. These cells generate the extracellular fibers and secrete a substance called hyaluronan that causes the ground substance to be viscous. Tissue macrophages are large, amoeboid-like cells that are scattered among the fibers. These cells engulf damaged cells or pathogens that enter the tissue. These cells play an important role in mobilizing the body's defenses, as you will learn later in our blood chapter. When stimulated, these cells release chemicals called cytokines that act as an attractant that causes more immune cells to come fight an infection. Adipocytes are also known as fat cells or adipose cells. A typical adipocyte contains an enormous lipid droplet. The nucleus and other organelles are squeezed to one side so the cell looks like a ring with a jewel on one side. Mesenchymal cells, again, are stem cells that can differentiate into other types of connective tissue cells. Wandering cells include leukocytes responding to tissue macrophage signals that arrive from the bloodstream along with mast cells. Some of these immune cells, called leukocytes or white blood cells, are found in connective tissue and include neutrophils, B cells, and T cells. Again, you will learn about these leukocytes in more detail during our blood chapter, but for right now, just realize that these are cells important for fighting infections. Mast cells are small mobile cells often found near blood vessels. The cytoplasm of a mast cell is filled with secretory granules of histamine and heparin. Histamine causes blood vessels to dilate or open up in diameter, and this increases blood flow. Heparin is an anticoagulant and thus helps maintain blood flow to an area. You should realize that these two chemicals allow more blood flow to an area of infection, and this means more leukocytes that arrive to help an infection. 3 types of fibers are found in connective tissue: collagen, reticular, and elastic fibers. Fibroblasts form all 3 types of fibers. Collagen fibers are long, straight, and unbranched. They are the most common and the strongest fibers in connective tissue proper. Each collagen fiber consists of 3 fibrous protein subunits wound together like the strands of a rope or a cable. Like a cable, the collagen fiber is flexible and very strong when pulled from either end. This kind of applied force is called tension, and the ability to resist tension is called tensile strength. Much like the cables of a bridge help support the weight of the cars and framework of the bridge. Reticular fibers, reticulum means network, contain the same protein subunits as collagen fibers, but they are assembled differently. Reticular fibers are thinner than collagen fibers, and they form a branching, interwoven framework that is tough and flexible, much like a sponge. These fibers are abundant in the spleen and liver, and they create a complex 3-dimensional network, or stroma, that supports the parenchyma, that means the functional cells of an organ. Because reticular fibers form a frame-like structure, they resist many forces from many different directions. Think of how you can twist, press, or squeeze a sponge, and thus afford organs much stability. Elastic fibers contain the protein elastin. Elastic fibers are branching and wavy and allow for stretching, but their most important function is their ability to return to their original dimensions. In other words, It is their recoiling properties that are most important. Think of this. If you were to wear your favorite underwear every day, with washing of course, one day you would reach into the dryer and your underwear would one day be the size of a moo-moo. You now slip this very large pair of underwear on and it immediately slides off your waist and back down to your ankles. What do you think? I bet you think, oh darn, the elasticity has worn out. So even though the elastic fibers have stretched greatly, they are now broken and have lost their recoil ability. So elastic fibers are important for stretching, that's true, but their ability to recoil is more important, and you will appreciate this ability even more when we talk about the respiratory system. Of the loose connective tissues, areolar tissue is the least specialized connective tissue and thus considered the, quote, model. This tissue, shown here as both a cartoon drawing and as a photomicrograph, contains all of the cells and fibers found in any connective tissue proper. Areolar has an open framework, and ground substance accounts for most of its volume. The ground substance cushions, shocks, and because the fibers are loosely organized, areolar tissue can be distorted with extra volume without damage. The elastic fibers allows areolar tissue to expand and return to its original shape after the pressure is relieved. In addition to carrying oxygen, nutrients, and waste, the blood vessels found in areolar connective tissue carry white blood cells that can then exit the vessel and enter the areolar connective tissue to fight infections. Additionally, the loose areolar connective tissue can hold or store nutrients in the form of lipids